In this video, we will discuss about how these mechanotransductions and innate immune activations are related. So, whenever there is some kind of bacteria that is interacted with this toll-like receptors, then it will trigger some of the intercellular signalings, such as NF-kappa beta 1 activation. At the same time, it will also open up the receptors such as piezo 1 that will increase the Ca2 plus influx inside the cells. And along with this, bacteria interactions with the toll-like receptors, sometimes the ECM also get changes, or its stiffness of the ECM gets changes because of the tumor microenvironment developed by the cancer cells, and that will cause these integrins to interact with the actin and cause these polymerizations of these effectins through the utilizations of this G-actin, and this will also cause this opening of this receptors, that is piezo1 TRP V4, that will increase the Ca2 plus influx, and this together polymerizations of these effectins and Ca2 plus will leads to this growth GTPase upregulations pathway, and simultaneously it will also increase phagocytosis. And since these G-actins are being consumed for the polymerizations, this MRTFA gets free and it will localize into the nucleus and interact with this SRF, and thereby triggering the genes that will cause upregulations of the cytokines such as interleukin 6, or we can say the genes that are responsible for this innate immune activations gets upregulated. And this link is, although it is not been proved that it plays a role in the innate immune response, it is a hypothesis that it interacts with the innate immune response, or it helps in the innate immune activations. And since this piezo gets activated either through the ECM stiffness interactions with the stiff ECM interaction with the integrin, or through this bacteria gets detected by these toll-like receptors, it will cause Ca2 plus influx. It will increase the Ca2 plus influx, and it will cause EDN1 and hypoxia, HIF1 alpha signaling pathways, and both of them will also upregulate the genes that will promote metabolism and enhance effector functions. And with these interactions of this high stiffness that we have discussed in the previous video, that now the YAP-TAZ complex cannot be that we have discussed in this. Whenever they interact with the stiff environment, the YAP cannot be phosphorylated, and it can go inside the nucleus or transloaded inside the nucleus, and same things will happen, that is YAP-TZ cannot retain in the cytoplasm, and it will enter inside the nucleus, interacts with the TAD that we have discussed, that it will interact with this here YAP-TAZ it cannot be phosphorylated, and it can interact with this TAD in the nucleus, and this TAD in the nucleus, then it will cause epigenetic changes in a way that will promote this glycosylase, pentose, phosphate pathways, TCA cycles, and also enhanced effector functions that are mentioned here. And through this TRP4 Ca2 plus influx, it will also affect this duplex DUSP1, and that will affect this P38, and that will also cause the upregulation of the phagocytosis. At the same time, it will inhibit this JNK pathways, which is somewhat related to the this cell death pathways. And in terms of these effector functions, it will lead to the upregulations of the cytokines that I mentioned here, that is interleukin 1 alpha, interleukin 1 beta, interleukin 6, TNF, CCL2, and I think these are the things that are mentioned here. Yeah, this HDACs, through this Ca2 plus influx, it will also affect the HDACs in the nucleosomes. But all these things that are been marked here as dotted arrows are not been come to a general consensus, whereas all this will be marked as dark line are been well accepted by the research community, or there has been general consensus. But in the dotted arrows lines, there is no general consensus yet, or the general consensus are yet to be established. So this is some of the interactions or how these mechanotransductions can cause these innate immune activations. If you need to understand more about this relationship between mechanotransduction and innate immune activations, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.